Hello, my name is Ashley Sue, and I'm a senior in the Math Science Technology Center at Dunbar High School. And I worked under the mentorship of Dr. Eileen Lin at the University of Kentucky. And my project is the study of sex and genotype-driven disparities in vascular integrity, metabolic profile, behavior, and the gut microbiome in Alzheimer's disease. So first, a brief overview into what I'll be doing and talking about today. So first, I'll talk about the introduction and background about what Alzheimer's disease is and what each individual function that I'm studying is. Then I'll go into the research objectives and the general goals for my project. Then I'll go into the methodology and then the results and discussion. And finally, I'll talk about the summary of my results, paths for further research, acknowledgements, references, and also ways that you can contact me in case there are any questions that you may have about my research. So first, a brief introduction and background on Alzheimer's disease. So Alzheimer's disease is a neurodegenerative disease that is characterized by cognitive impairment and also dementia. And so while all cases of Alzheimer's disease have these two major hallmarks, what's unsure of in the scientific community is the exact mechanisms in which each individual develops Alzheimer's disease. Alzheimer's disease is a deeply personal disease, and many people may have different mechanisms and different symptoms leading up to it. And so some of these major hallmarks might include the inflammation of the brain, and aggregation of tau tangles, and also amyloid beta plaques. And so these are some of the molecular technicalities that begin to aggregate in the brain in developing Alzheimer's disease. But in any case, Alzheimer's disease, what we're seeing is that there are some major patterns in the development of Alzheimer's disease in different demographics of people. And that is something that I wanted to study in my project. And so the two major risk factors that are associated with Alzheimer's disease include the genotype risk factor and also the risk factor associated with gender. And so out of all of the genes, the main gene that has been identified that contributes to Alzheimer's disease is the ApoE gene, or the apolipoprotein E gene. And so this gene comes in two alleles, the ApoE3 gene and also the ApoE4 allele. And so these two alleles have vastly different effects on the person. And so the ApoE gene has been identified to majorly affect the two major hallmarks of Alzheimer's disease, neuroinflammation and also amyloid beta protein buildup inside the brain. And so what the ApoE3 gene is the normal unmutated version that has been seen in humans. And the ApoE4 gene is a mutated version of that gene that increases the risk factor for developing Alzheimer's disease. Someone with one copy of the ApoE4 gene has about 3 times the risk of someone with the ApoE3 gene, versus someone with 2 copies of the ApoE4 gene has upwards of a 12 times risk of developing Alzheimer's disease. In addition to this gene, also a major factor in determining the risk factor for Alzheimer's disease is also the gender of the individual. And so what you can see is that there is a larger instance of Alzheimer's disease in women compared to men. Women have about a 2 times higher risk of developing Alzheimer's disease compared to men, just for the fact that they are women. And so one thing that is really interesting is just that this figure actually can't just be explained by the fact that women tend to live longer than men, and their average lifespan is a little bit longer. And so even though there is this trend, we cannot attribute that entire doubled risk factor to just the fact that we see that women live a little bit longer. And so these two major risk factors affect the person differently and obviously change their risk for developing Alzheimer's disease. And so the main goal of my project is to figure out exactly what about that ApoE gene, or what about being male or female, is contributing to that increased or decreased risk factor. Something else that is really interesting is that the ApoE gene seems to influence women a lot more than it does men. So women who have that ApoE4 gene have a significantly increased risk factor for developing Alzheimer's disease compared to men with that ApoE4 gene. And we also see that women are far more protected by that ApoE3 gene compared to males. And so for some reason, this ApoE gene is also more effective on females, whether that is protecting them or causing them harm. And so the main goal of my project is to be able to understand that mechanism to lead a path for future precision medicine. And so precision medicine is really important just because we see that we need to have a preventative therapy for developing Alzheimer's disease. Because Alzheimer's disease is both progressive and neurodegenerative, we need to make sure that we are able to create preventative therapeutics so that these treatments are more effective for Alzheimer's disease. Rebuilding brain tissue isn't exactly an easy task, so being able to know exactly what we need to target before Alzheimer's disease manifests is a really large goal of my project. And so being able to better understand the mechanisms of both women and ApoE4 individuals developing Alzheimer's disease will allow for more precise therapies, and knowing exactly what we need to target will help larger demographics of people be able to figure out what they need to treat Alzheimer's disease, especially in a preventative manner. And so the methodology that I used, because I'm trying to find those baseline differences, I used control data from previous studies. So there have been a lot of studies conducted that have been looking at specific pharmaceutical or dietary preventions of Alzheimer's disease. And because I'm not really looking for a specific um, treatment or a specific therapy, I can just use the control data from previous studies that have done so. And so because I'm looking at this baseline data, because what I'm doing is so general, I was able to find lots of different metrics for measuring Alzheimer's disease progression. And so the four that I focused on was vascular integrity, behavior, metabolic profile, and gut microbiome. And so the vascular integrity is really important just because we do see that neurodegeneration in Alzheimer's disease. So knowing how well the blood vessels inside the brain are able to um, hold up, especially near the end or near more middle-aged periods of people's lives, is really important. The behavior is exactly what it sounds like. It's just something that you see as a direct result of Alzheimer's disease, is the behavior of an individual. The metabolic profile is really important because each individual metabolite is a byproduct of a specific process in the brain. So being able to accurately determine the profile of the metabolites inside the brain is really indicative of the exact processes that are happening. And increasing literature has shown more and more correlation between the gut microbiome and the brain. And so knowing that those baseline differences in the gut microbiome is really, really important, especially if we go through a future of more dietary preventions for Alzheimer's disease, as those dietary preventions directly go into your gut. So knowing what's happening there and knowing that there is a link between the gut and the brain, I thought it was important to also look at the gut microbiome as well. So what I did was, after I took that data from other studies, I put them into a program called GraphPad Prism, where I conducted statistical tests to determine exactly what was different. And so mostly what I did was I used Welch's two-tailed t-tests to determine the p-value, to determine whether or not those differences were actually statistically significant or if they were just different by pure chance. And so one thing that I did additionally to those t-tests was the false discovery rate test. And so while I was able to do these Welch's two-tailed t-tests for both the behavior and the cerebral blood flow, I had to do an additional test, this FDR test, because both the gut microbiome and the metabolic profile, those data sets were very, very large. And so when there was over 500 metabolites, even that p-value of 0.05, that 5% chance that it is by chance, isn't exactly strong enough to say that out of those 500 metabolites, so many are significant, because even in 500, 5% is still a lot of metabolites. So I wanted to make sure that I was finding exactly statistically significant results, and that was something that I could do using that false discovery rate. And that was also the same situation with the gut microbiome. And so there are thousands of different species of bacteria in the gut. And so using that false discovery rate is really, really important when discovering those differences. So the results of my project actually is really, really interesting. So the metabolic profile, a lot of the disparities were actually driven by that genotype, the ApoE3 versus ApoE4. And so out of the ones that were significant, we can see here that because of the Q value has to be because the Q value has to be below 0.05, it drives down the P value really, really low, so that I know that these 5 metabolites are significant, and there's no question about that. And so, actually, these 5 metabolites have really interesting stories behind them. So, N6-methylysine, according to literature, actually is involved. Actually, if found on the tau protein, can actually reduce tau protein aggregation, which is one of the hallmarks of developing Alzheimer's disease. And so having that aggregation and knowing that one specific metabolite is actually responsible for that reduction of aggregation is really important, especially as a lot of the Alzheimer's therapies have overlooked this N6-methylysine metabolite. And so while we do know that, well, literature has shown that this specific metabolite can be involved in that reduction of aggregation, no therapies have used that as a target for their therapy. And so this is a great example of somewhere where I could go in for future research, just because it's something that hasn't really been studied yet and can really lead to a lot of different possible target plans. And so something else actually that is really interesting is this N-acetylarginine. And so I'll do this in the graphic. So what we see here is that N-acetylarginine is actually, there's higher levels in females who are ApoE3 compared to ApoE4. And there's also higher levels in ApoE3 males compared to ApoE4 males. And so acetylarginine actually has a lot to do with oxidative stress. So acetylarginine is a marker that is a byproduct of oxidative stress. And so this is actually really interesting, because oxidative stress is also another hallmark of Alzheimer's disease. And so someone with lots of oxidative stress has risk of developing a lot of damage inside their brain because of this. And so it's really strange how something that is supposedly the baseline or normal version of the ApoE genotype actually has increased markers of oxidative stress, which is something actually that is really interesting, because when looking at the next metabolite, 3-uridyl-propionate, it's something that is also significantly different when looking at males versus females. And so what we see is out of all of those metabolites, out of the 500 something metabolites that I looked at, these were the only 2 that were significant. And so there was the 3-uridyl-propionate and the pantothenate. And something that's really exciting about these 2 is the fact that they're both linked to the development of vitamin B5. So pantothenate actually is the scientific name for vitamin B5, and 3-uridyl-propionate is actually involved in the production of one of the major building blocks of pantothenate. And so knowing this, what we see here is that these females have significantly higher levels of pantothenate and uridyl-propionate compared to males. And so one thing that's really interesting is that this pantothenate actually is an antioxidant. And so knowing that these females, these ApoE3 females specifically, have higher levels of this antioxidant compared to males may explain why we see that those who are ApoE3 still have higher levels of that acetylarginine, which is an indicator for oxidative stress. And so what I'm proposing is that a possible mechanism for why these ApoE3 females are so much more protected from Alzheimer's disease compared to ApoE3 males is that presence of pantothenate in that pathway. And knowing that there is one specific antioxidant that might be the contributor to that protective measure for those female mice who are ApoE3 might be an indicator as a possible therapy. And also knowing that this vitamin B5 is something that you can find on your cereal boxes or find in the pharmacy is also really compelling, just because those who are not, for example, ApoE3 females, might be able to do some more of their preventative therapies through supplementation of pantothenate or vitamin B5. And so knowing that there is one specific antioxidant that might be contributing to the protection in Alzheimer's disease is actually pretty exciting. And so on top of the metabolic profile, I also looked at the vascular integrity and behavior. And so one thing specifically that was really interesting is that these ApoE4 females had significantly worse vascular integrity compared to both males who were ApoE4 and also females who were ApoE3. And so the study that I obtained the data for vascular integrity actually measured the vascular integrity through the cerebral blood flow at both 4 weeks and 16 weeks. And so 4 weeks, the mice are a little bit younger. This is still when the mice are pretty young in age. And then 16 weeks is a little bit younger than middle-aged if we were to convert that to human years. And what we see is in both situations, these female ApoE4 mice have significantly lower cerebral blood flow in the hippocampus. And so we see that this is the case in both younger and older mice, showing that even though these mice were younger, they're still at a higher risk for just a reduction in their blood flow, especially in the hippocampus, which is especially important because the hippocampus is the learning center of the brain. So knowing that they're not getting as much blood flow, they don't have that vascular integrity inside their hippocampus is really indicative of the next section that I looked at, which is behavior. And we see a similar pattern in that these ApoE4 mice can have significantly worse performance. In both graphs, they have larger errors, larger numbers of errors compared to the respective male mice or the ApoE3 mice. And so what's really important to actually is that this is happening in the third block. And so the radial arm water maze, the way that it works is the 6 blocks are conducted over 2 days. And so the first 3 days is really the learning phase, where the mice are first exposed to this radial arm water maze, and they have 3 trials to begin to understand exactly what's going on. And then the second 3 days is more of a recall phase. So having this kind of test that tests both the memory ability as well as the learning ability is really indicative of what's happening in these brains. And so what we're seeing is both of these significant differences are happening in the third block, which is the final learning phase. And so knowing that there is a specific application and there is a specific result of that decreased vascular integrity is really important, especially when looking at the learning ability of these mice. So this is really something that is really useful, especially when looking at therapies, knowing that if someone is ApoE4, maybe supplementing with that antioxidant might not be the best approach to treating Alzheimer's disease or preventing it. And we might be in the scientific community may be more motivated to look for more vascular approaches and preventing that deterioration of the blood vessels inside the brain. And then finally, I looked at the gut microbiome. And so this is the alpha diversity in the gut. And so alpha diversity is essentially just the number of different species inside the gut, and knowing if there is a very diverse area or if all of the microbes are limited to just a couple of species. And so what we can see here actually is that there is a significantly lower level of alpha diversity in these ApoE4 females. And so it's significantly reduced compared to the males in that ApoE4. And so this is actually a trend that is also similar to those of obese patients. And so those who struggle with obesity actually have a lower alpha diversity. And so seeing this correlation between Alzheimer's disease and also the obesity is extremely important also in knowing the different correlations between the different correlations of different diseases, which include Alzheimer's disease, and knowing that there is a large impact of diet on the brain in seeing that this genotype, which is largely affected, which largely affects Alzheimer's disease. Also, we're seeing that these ApoE4 females have significantly lowered alpha diversity in their gut. And so just a summary of the results that I had. So the panthotenate and 3-uretopropionate are essentially the antioxidants. Of the metabolites, these are the 2 that are significantly changed by the gender of the individuals. And so actually 3-uretopropionate, as I said, is both influenced by the gender of the individual and also the genotype of the individual. And so this is the mechanism that I'm proposing, is that they're able to combat the negative effects of the N-acetylarginine, which is a marker of the neuroinflammation and oxidative stress. And so this is something that is that is at a higher level in those ApoE3 mice, which are supposedly protected from Alzheimer's disease. And then N-methylysine is actually something that reduces tau protein aggregation and may be a very interesting pathway into future developments for Alzheimer's disease therapies. And homocarnosine actually is the neuronal derivative of carnosine. And so it's associated with amyloid beta clearance and is also something that is associated with, that the disparities are driven by the ApoE gene. And then out of vascular integrity and behavior and the gut microbiome, in both of these situations, we're seeing these ApoE4 mice, these female ApoE4 mice have significantly worse performance than any of their peers. So the males and the ApoE3 females are all doing significantly better than these ApoE4 females. And so knowing this is really indicative of what kind of treatments we might be looking for in these females. Which brings me to these further research. So a lot of the elements of my project is really specific and really indicates the specific mechanism and the specific processes that are happening inside the brain. So knowing these mechanisms and knowing these pathways for developing different aspects of these different hallmarks of Alzheimer's disease is really important and really influential in being able to figure out what exactly we need to target with Alzheimer's disease. And so this, because I also looked at that gut microbiome, it's really important to see that what I'm looking at is not limited to just pharmaceutical interventions. It's also something that can be open to different studies on different dietary pathways of treating Alzheimer's disease. And so knowing that the applicability of my research in people's everyday life, I think is really, really important. And knowing that just being able to change your risk for Alzheimer's disease much before you're even thinking about Alzheimer's disease, I think is really important, especially as we do have an aging population. And so I would like to acknowledge a couple of people. First of all, my mentor, Dr. Eileen Lin, for guiding me and providing me with the resources to do my project. I would also like to thank Ms. Young from the MSTC program for making sure that this program is available to us and allowing us to do our research during the school year. I'd also like to thank KYRP, or the Kentucky Young Researchers Program, for allowing me, a high school student, to be able to do university level research at the University of Kentucky. And finally, I'd like to thank friends and family for the support and the rides that I got to lab. So these are my references that I referenced inside this PowerPoint specifically. And if you have any questions, feel free to send any to my personal email, ashley.sue@stu.fayette.kyschools.us. Thank you.